Everyone and welcome to my channel AG Life Science. If you like my video, please subscribe my channel and share it to your friends. Press the bell icon for notifications. Here in this lecture, we are going to discuss about the electron transport chain. The electron transport chain is a ATP generating process in a metabolism. We know that in a glycolysis, the net gain of a ATP is 2 ATP molecules. In a Krebs cycle, the net gain of ATP is a 2 ATP. But the 4 ATP molecules does not fulfill the requirement of energy. So there must be another way for the ATP generation. There is a better way, that is electron transport chain. Electron transport chain is situated in an inner mitochondrial membrane. It contains a number of carrier molecules, and these carrier molecules are arranged in a series. They are associated with a cristae of a inner mitochondrial membrane. They are nothing but a transport proteins and associated with some enzymes. A transport of electron through a electron transport chain is linked to a pumping of H plus ions, and it creates a H plus gradients across a inner mitochondrial membrane. And the yield of ATP molecule by a complete oxidation of one glucose is 34 ATP, and this breakdown occurs in only presence of oxygen, so it is aerobic respiration. In aerobic respiration, there is a need of oxygen. Here the final electron acceptor is a oxygen. When the electrons from the EMP pathway and the TC cycle are run from a ETC, the electron lastly reach to the O2 and leads to formation of a water molecule in a aerobic respiration. But in anaerobic respiration, the final electron acceptor is rather than a oxygen molecule. It may be a carbon dioxide or it may be a SO2, that is a sulfur dioxide. The organelle in which there is a presence of ETC is a mitochondria. The mitochondria is made up of a double membrane, or it is made up of two membranes. One is a outer membrane and another one is the inner membrane. The inner membrane, it is a highly folded structure, and this unit of inner membrane is a cristae. And this cristae or inner membrane is associated with enzymes and transport proteins. In between outer membrane and inner membrane, there is a one space is called as a intermembrane space, and here is a fluid in between the intermembrane space. The electron transport chain, here is the electron transport chain. This is a inner mitochondrial membrane. Here this is a mitochondrial matrix. This is a intermembrane space. It is associated with a number of complex The first complex, that is a NADH dehydrogenase, second complex, that is a cytochrome BC complex, third, that is a cytochrome C oxidase complex. The electrons are gained from the different metabolic pathway from a EMP pathway or a TCA cycle, where at this pathway where the substrate get oxidized, it raise out a hydrogen or a electron molecule. Number of reactions are there in a glycolysis and in a TCA cycle, that the substrate get oxidized, and this oxidation of a substrate releases a hydrogen molecule or a electron molecule, and this hydrogen molecule or electron molecule get transferred from a ETC. So here are the some steps. We know that the one step is there where glycerol 3 phosphate is get dehydrogenated to the 1,3-diphosphoglycerate, and this is a dehydrogenation reaction where there is a release of a hydrogen molecule. Also, when the pyruvate leads to formation of a acetyl coenzyme A before entry into a TCA cycle, it also get dehydrogenated, and this removed hydrogen is takes part in a ETC, or it get transferred from a ETC. In a TCA cycle, there are the 3 steps where there is a removal of a hydrogen molecule or a substrate get oxidized. Mean the substrate for these hydrogen or electron are NADH or a FADH. They can take up the hydrogen or electron. Now, the electron transport chain, as said earlier, the hydrogen molecules which are released from a different metabolic pathway like EMP pathway or a TCA cycle, this hydrogen molecule or a electron molecule get transferred from a number of carriers which are situated in a inner mitochondrial membrane, and these all carriers totally called as a electron transport chain. It is made up of a number of complexes. These are NADH dehydrogenase, cytochrome BC complex, and cytochrome C oxidase complex. These complexes or these carriers are located on a inner mitochondrial membrane, and here this part is a mitochondrial matrix and this part is a intermembrane space. The electrons flow downhill. It flows or it gets moved into a stepwise manner, and it is finally accepted by a molecular oxygen. Each carrier or each transporter is a electronegative. Here there are some carriers. First is a FMN, that is a flavin mononucleotide. It is with a NADH. The next carrier, that is a FES. Then Q, that is a ubiquinone, cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and A3. These are the 3 complexes: complex 1, complex 2, complex 3. A complex 1 is made up with a NAD and FMN and APS. The complex 2 is made up of cytochrome B and cytochrome C1. Complex 3 is made up of cytochrome A and cytochrome A3. Also, here is one pump. It is called a ATP pump because of a movement or a flowing of a hydrogen or a electron. There is a formation of the H plus ion gradient across the inner mitochondrial membrane, and these gradient or these H plus ions get flow back into a matrix through a one transporter, and this transporter is called as a ATP synthase transporter or a ATP pump, and this allows the flow of a proton, and because of this flow of proton the ADP molecule get phosphorylated into a ATP. So this is a story why the ATP are produced in the electron transport chain. The electron transport chain couples a chemical reaction between an electron donor and electron acceptor. Each and every transporter firstly get reduced and simultaneously it get oxidized by donating an electron to a next carrier. So it leads to a formation of a H plus ion gradient across a membrane, and these H plus ions are further used for a synthesis of a ATP. ETC is used for extracting energy from a sunlight through a process of photosynthesis in a plant, and from a redox reaction such as oxidation of sugar like a glucose, and this is called as a respiration. Here is also a phosphorylation reaction and ATP is produced. The glucose get converted into a pyruvic acid through a process of glycolysis. Further, the pyruvate is get converted into a acyl coenzyme A, and it is transported into a mitochondrial matrix, and there is a running of a Krebs cycle. The electrons from a glycolysis are present in a cytosol, and these are carried through a NADH. Also, the electron from a Krebs cycle also carried by a NADH and a FADH2, where these electrons are used for a synthesis of a ATP through a oxidative phosphorylation. You know that in a Krebs cycle there is also formation of a ATP molecule, but this ATP molecule is formed through a substrate level phosphorylation. Succinic coenzyme A is get converted into a succinic acid. This is one step where there is a generation of a ATP by a substrate level phosphorylation. Now the components of the ETC, the NAD and flavoproteins. These are the hydrogen carriers in a cellular respiration. A second component, that is a non-heme metalloprotein, that is a FES protein, and these FES proteins are with a ferrous ion, and they get cycled into a 3 plus and 2 plus state when this metalloprotein or a FES protein accept the electrons from NAD or a FAD or FMN. They get reduced in a 2 plus state. Simultaneously, they donate an electron to a ubiquinone and get oxidized to a 3 plus state. The electron from oxidized FES protein is accepted by a next carrier in a series, that is a ubiquinone or a coenzyme Q, and this ubiquinone or a coenzyme Q is serves as an anchor to a inner mitochondrial membrane. Then next to ubiquinol or a cytochrome coenzyme Q, there is a presence of a carrier, that is a cytochrome. And this carrier or this electron transport protein contains a heme and a phosphatic group. They may be a C1, C and A and A3. Now it is about the composition of a ATC. The ATC is composed of four large protein complexes. The complex 1 is with a NADH and coenzyme Q reductase. Complex 2 is with a succinic acid and coenzyme Q reductase. Complex 3 is with a cytochrome C reductase, and complex 4 is with the cytochrome C oxidase. The many of the components are a protein in nature which show a prosthetic group to move the electrons through a ATC. The first component is a NADH dehydrogenase. The electron pass from a NADH or bypassed from a FMN, that is a flavin mononucleotide, to the APS cluster and to the ubiquinol, that is a coenzyme Q. During this transfer, the APS get reduced and it get oxidized simultaneously by donating the electron to a ubiquinol or a coenzyme Q. Then the complex 2, that is a succinate dehydrogenase, it is an entry point for a FADH2. The succinate dehydrogenase, we know that the succinate dehydrogenase is a enzyme of a TCA cycle, and this enzyme directs the formation of a succinic acid via FADH2. The next acyl coenzyme A dehydrogenase. This enzyme is from a beta oxidation of a fatty acid, and it also transports electrons to coenzyme Q via FADH2. It means a succinate dehydrogenase and acyl coenzyme A dehydrogenase are resembling to each other. Then the complex 3, it is with cytochrome B, C1 and C. And this complex 3, they transport a electron from ubiquinol to a cytochrome C, and these cytochromes having a heme as their phosphatic group. Then the complex 4, is with a cytochrome A and A3. It is made up of a 10 protein subunit. Two types of phosphatic groups are here, that is one is a heme and another one is a copper. Electrons from a complex 4 are delivered to a O2. Several chemicals can inhibit this pathway. Majorly, the cyanide and a carbon monoxide can block electron transport between cytochrome A or a cytochrome A3 to a molecule oxygen. So the cyanide, because of this inhibition ETC, it is poisonous. Also the carbon monoxide, it is also poisonous because it inhibits electron transport through a ETC at a complex 4. This is about the flow of electron. Complex 1st bifurcated with a complex 2nd, complex 3rd, complex 4th. This is a outer mitochondrial membrane. This is the inner mitochondrial membrane. The electron, or there is a saturation of a hydrogen in this intraphase. This is a intraphase, a phase between two membranes. The hydrogens are saturated in this area and get back return into a matrix across a inner mitochondrial membrane. But in a inner mitochondrial membrane, there is a ATP synthase enzyme and it leads to formation of ATP by using ADP and one inorganic phosphate. So there is a need of a H plus gradient across a inner mitochondrial membrane, and the energy stored in a proton form is utilized for a production of ATP. Now the functioning of ATC. In a ATC, the carrier molecules are arranged in an increasing order of their redox potential and a decreasing order of their electron pressure, diminishing the free energy levels. The terminal electron acceptor for aerobic respiration is a molecule oxygen. As the electron flowed from a ATC, it loses its free energy. In a ATC, enzyme bound H plus is used as a fuel for energy generation. In oxidative phosphorylation, the electron moves from an electron transport chain from ADH and FADH to O2, means where ADP is phosphorylated to ATP. These two processes of electron transport and formation of ATP are a dependent process, and they are dependent on each other. ATP cannot synthesize unless there is energy from electron transport, that is 31 kg per mole. Electrons do not flow to O2 unless there is a need of ATP. 3 ATP are generated when 2 electrons are transported from NADH to O2. 2 ATP are generated when 2 electrons are transported from FADH to O2. Thank you. If you like my video, please subscribe my channel, share it to your friend and press the bell icon.